Thank you, David. Good afternoon, everyone. So I'm Robert Nash from the United Kingdom. We have laboratories based in Aberystwyth, shown by the green dot there on the map of the UK. We're in the west of Wales by the sea. And we've been working on some plants with Professor Cartereri from Africa and also other places. And I wanted to give you a very quick overview of some of the work we've been doing in the past that's led into this current study on African plants and food products. So the title of my talk is Amino-Sugars in African Plants and Their Benefits to Health. Amino-Sugars, for those of you who are not familiar with them, are probably extremely common plant and microbial natural products, but they're very rarely studied because they're very water soluble. And by tradition, that a water extract is very difficult to work with. Often you have large amounts of primary metabolites like the sugars and amino acids, and this means that separating interesting molecules from that water-soluble fraction can be very difficult. And very often the active compounds are heavily diluted by the primary compound. But nonetheless, there are some very interesting compounds in there, such as amino-sugars, and hopefully I can get you interested in the amino-sugars of plants and microbes. And it was very interesting to hear about the interest in water-soluble extracts from the mushrooms from Dr. Sokoka earlier. I think the water-soluble fractions of a lot of products are very worth having another look at. So what are amino-sugars? Well, in their simplest form, they're analogues of sugars which have the oxygen in the ring replaced by nitrogen. There are a number of different forms these can take in plants and microorganisms. They can be very simple analogues of sugars like glucose, sucrose, monocyclic sugars, or they can be bicyclic compounds, resembling often disaccharides. But they tend not to be much bigger in plants. The interesting thing about them is they're very stable. Obviously, they're very small. They're selective glycosylation inhibitors or receptor binders. Very orally available, probably actively transported, as would sugars be. Metabolically inert, and they tend to go around the body, interact with enzymes and receptors, and then excreted completely unchanged, which is really quite surprising. And the other reason that very few of these have actually been known or studied in the past is that the chemistry is quite complicated. Organic chemists have also traditionally not liked working with very water-soluble compounds because it slows down their reaction processes, and their separation techniques are often not suitable for working with very water-soluble compounds. And so very few of these compounds had actually been synthesized until we started collaborating with a group at Oxford University who synthesized a number of the natural products we produced and then variations on the theme. And this enables us to prove the structures, which can be quite tricky because of the number of stereochemical centers. So the first natural amino-sugars were noduramycin and 1-deoxynoduramycin, discovered in the 1960s and into the 1970s. So noduramycin is a bacterial product produced by Streptomyces and Bacillus species in particular, but there are probably many other bacteria which also produce amino-sugars which haven't been discovered yet because, again, they're difficult to find in the culture media. The broths are often quite complicated with a lot of culture nutrients, which make the discovery of the amino-sugars quite tricky in amongst that big mess you get after a fermentation. 1-deoxynoduramycin was found in mulberry leaves and was found following Japanese workers looking at the traditional use of mulberry for treating diabetes, and they found this compound, 1-deoxynoduramycin, which is a glucosidase inhibitor and has formed the basis of a number of anti-diabetic drugs which work on digestive disaccharidases and control therefore blood glucosidase levels by slowing the uptake of glucose from the gut. But there are a number of side effects which this activity causes, such as diarrhea, stomach cramps, which makes these drugs not ideal as treatments for diabetes. And now coming to some of my own work. This was on African plants. As part of my PhD, I worked on this plant, Angelicalyx brownii, which occurs in the north of Africa, around Kenya, Tanzania. And we found it contained this simple amino sugar compound, which was a new structure, and it's an analog of fructose, but it's a potent inhibitor of alpha and beta glucosidases. Since discovering it, it's carried on to be worked on by other groups as an inhibitor of glycogen phosphorylase, a number of other glucosidases, and we also showed that it had an ability to inhibit the formation of active HIV particles in vitro. And it's currently being modified also as a treatment for Pompe disease, as a chaperone for deficient glucosidase involved in the lysosomal storage disorder Pompe disease, which is a big problem in children and is very difficult to treat. So, talking a bit about the anti-HIV activity of these molecules, from the compound from Angiocholic brownia, we also discovered that other glucosidase inhibitors, such as casnospermine, the bicyclic molecule there from an Australian legume, and also 1-deoxynitromycin from mulberry, also had an anti-HIV activity. And we were able to show via funding from the Medical Research Council in the UK that what these glucosidase inhibitors do is they affect the ability of the HIV virus and many other viruses, which are glycosylated, to form their surface glycoproteins correctly. So what these molecules do is they inhibit various trimming reactions, which are there to essentially ensure that glycoproteins are produced with the correct structure. And the virus needs to produce particular glycosylation patterns on GP120, for example. And in the presence of these inhibitors, it can't form GP120 correctly. And so the virus is produced, but it's not infective. So it's a very good way of making HIV and other viruses non-infective by changing the glycans, the glycoproteins, which are expressed on the surface of the virus particles. And this is still a novel approach for tackling viral infections where the viruses have surface glycoproteins involved in the infection process. And hepatitis C is another potential target for these types of compounds, which is still being studied. So following on from the discovery of these amino-sugars, which have activity through being analogues of carbohydrates, which are important all around the body. I mean, it's not just virus particles that have glycoproteins on the surface. All our cells have glycoproteins on the surface. And these glycoproteins are very important to how cells function and interact with each other. And very, very important, for example, for how the immune system functions and picks up aberrant cells and infectious particles. And this, because of this great complexity of carbohydrate structures in the body and the structural and biological importance of these carbohydrates, opens up tremendous opportunities for finding new drugs in the future. And we think that the activities of many herbal medicines are actually explained by their effects on carbohydrate processes in the body. And in fact, you find that many diseases, if not all diseases, have aberrations in carbohydrate metabolism, which can be picked up and are often used as markers of disease by blood sampling, for example. So elevation of particular enzymes like hexosaminidase or glucuronidase are often picked up as markers of disease, but we think they may actually be involved in the disease development as well. So we discovered, though, that glycosidase inhibition, so inhibition of glucosidases or mannosidases, which was the first activity noted for these molecules, is actually not the most interesting effects that these compounds can have on the body. So casearines was a new group of amino-sugars that we discovered. These are pentahydroxy bicyclic pyrrolizidine alkaloids, but they're not liver poisons like most pyrrolizidine alkaloids. These compounds modulate the immune response. And we discovered that one of these molecules was present in a herbal medicine from Western Samoa, and it was given to us by a patient who had been diagnosed with cancer, and a herbalist in Western Samoa gave her this dried powder. We discovered casearine as a new molecule. But then subsequently, we also discovered it occurred in a number of other quite rare medicinal plants around the world, which had very interesting claims for activity, treating AIDS, cancer, TB, leprosy, in fact, almost everything, and including one plant that occurred in Africa, Syzygium guianense, which many of you will know as waterberry. And we discovered that this active molecule casearine, in fact, as the casearine glucoside, can also be found in a rare honey from Africa, where it's concentrated by the bees. And the immune modulatory activity of this molecule is very interesting in that it stimulates the type 1 immune response, which is required for tackling cancers, TB, leprosy. In fact, many of the claims which were made for these plants around the world can be explained by the ability of these molecules to boost the type 1 immune response. And we've done a number of studies with immunology labs and also in mice and in people showing that this is indeed what this molecule can do, both in isolation, but also the honey from Africa can also do the same thing. So we discovered that, in fact, what this molecule does is it's not an enzyme inhibitor that's causing the immune modulation, it's a receptor binding. And we showed a particular receptor which occurs on immune cells, so macrophages, neutrophils, and dendritic cells, which interact with this molecule when it's eaten. And that leads to an increased activation of the immune system, leading to the ability of this molecule to have a number of disease healing effects, and also can have a prophylactic effect, preventing infection from viruses, for example. So the cancer application was what we were primarily interested in originally for the pure molecule, and we discovered that it has a number of interesting properties, rapidly orally bioavailable, can be synthesized fairly cheaply. A single oral dose given to mice with a metastatic melanoma can give a very pronounced improvement in the number of colonies of the melanoma which form. So in the middle there, you can see a lung on the left of the mouse where no treatment is given, and then a single dose of 100 microgram per kilogram of the pure molecule gives almost total clearance of the lungs from the metastatic melanoma after it's injected into the tail vein. The other interesting thing about this molecule and this group of molecules, the Imino-Sugars, is that they're not toxic. And so we think this opens up some very interesting new possibilities for anti-cancer drugs, for example, which are not toxins, which can have an anti-cancer activity, but which are not detrimental in the same way as most cancer patients, which are very often damaging to the immune system. So we've given very high doses to mice, up to 150 mg per kilogram, and shown no toxicity, whereas the doses, as we mentioned before, which are effective in mice, are actually well below 1 milligram. There are a number of plasma markers you can use, such as increased production of or responses to interferon gamma, for example, in response to various antigens. And we think it's interacting with a very small specific C-type lectin. So what it does, it also produces a very long-lasting improvement in the Th1 response. So an individual who's not responding effectively to a cancer cell can be given this molecule or the honey, and they show an increased immune response via activation of NK and T cells. And this leads to a reduction in the number of metastases in particular, which we've shown in mouse models, but we believe can also actually reactivate the immune system to tackle established tumors in animals as well. And because it's completely non-toxic, it could be completely safe to use in combination with surgery, chemotherapy, and radiotherapy. We've done a lot of studies showing the lack of toxicity. I won't go into detail there. We've done ADMET, DMPK, and biomarker studies showing that it seems to be completely safe with no toxicity. And this is a general feature, in fact, of the Imino-Sugars. The worst side effects you seem to get are those with compounds like deoxynitromycin, which cause gastrointestinal disturbance, which can be explained purely by their inhibition of digestive enzymes, which is not a very serious toxic effect, and you only see that at very high doses. You wouldn't see it certainly with micrograms per kilogram. So going on then to the discovery of this in a honey from Zambia, when Syzygium guianense, the waterberry flowers in Zambia, the bees obviously seem to have a plentiful supply of nectar, and they can produce honey which has a very high concentration of casein in the form of a glucoside. And so we can select from various drums collected over the year, find the drums which contain the casein glucoside and select those drums and then package it into a product, which we've done, and we call this amino honey. So each sachet has 5 mils of the honey with greater than 1% of the casein glucoside. And this is more than enough to give an increased type 1 immune response from an oral dose. And we're looking at marketing this product in combination with Professor Cartereri and also doing clinical studies looking at healthy aging, immune support, and also we know that the molecule also improves vaccine effectiveness, and so could be used, for example, to increase the effectiveness of vaccines and make those vaccines go further and therefore reduce the dose. This is another amino sugar. I'm just going to very quickly give some other examples of why we think these amino sugars are probably very common in African plants. Buckwheat is well known to contain this molecule, phagamine, which we discovered a long time ago. It's a weak glucosidase inhibitor compared to the oxynodromycin, but it does promote a more effective insulin response and lowers serum cholesterol, and seems to have some claims around improving energy levels. But this molecule also occurs in very high concentrations in a plant called Xanthocercis zambutica, which is obviously an African plant. And more recently, with Professor Carteret, we've discovered that some honey samples from Zimbabwe also contain this molecule, phagamine. So this could open up opportunities to actually develop a number of honey products for Africa containing amino sugars with known benefits to health, including potentially improving beta cell function and lowering insulin resistance, for example, even though quite surprisingly that occurs in a honey. And then another compound which also occurs in an African plant, but we first discovered it in Gymnema species, which is an Asian genus. Gymnema is well known to be a treatment for diabetes in Asia, proven to work in man in a number of clinical studies, but we feel the wrong compounds are being studied. Almost all the groups are studying a group of saponins, which have effects in vitro in cell cultures, but we don't believe they're that effective in whole animals. Whereas we've isolated this compound from Gymnema, which is an amino sugar acid, we called it BR1. And this molecule, in isolation given to mice, has the ability to control blood sugar levels. So we think the saponins are a red herring and are actually distracting people, in that saponins disrupt membranes, and you put them into a pancreatic cell culture and you'll get insulin released. We think in vivo, the important molecule is this single amino sugar acid, which is present at a very high concentration in some Gymnema species. Interestingly, we have found that this same molecule also occurs in some Baphia species, which is another legume species occurring in, or Baphia genus rather, a number of species which occur in Africa. But it occurs in Baphia in a much more complex mixture than occurs in Gymnema. But there is the possibility, we feel, of developing an African product based on extracts essentially of Baphia species. It'll be interesting to hear if anyone knows of any traditional use of Baphia species, for example, for controlling diabetes. We haven't found any in the literature, but maybe locally it's known that Baphia does have this ability, and it may be possible to separate out the active compounds from the compounds that are perhaps not so beneficial, develop an African product for diabetes, which may be better than Gymnema without the saponins, perhaps. So this is just showing that this molecule, BR1, works by oral dose and also by IP dose and also pre-dose in controlling elevated blood glucose after a maltose challenge. And the fact it can work IP shows that it's not inhibiting digestive glucosidases, and this means we feel it's a great advantage over deoxynodromycin, that it's not a glucosidase inhibitor and it doesn't need to go via the gut, and therefore you can avoid the side effects caused by DNJ by the inhibition of digestive glucosidases. These compounds are also much more widely distributed around the world. I'm sure there are many more examples to be found in African plants, but just as an example of another quite common plant that we've been looking at as a source of these and which we're developing products from is cucumber. Certain varieties of cucumber, and there are around 4,000 varieties of cucumbers now available. Certain varieties contain this compound, which we call IDO-BR1. It's got a different stereochemistry on the carboxyl group, and this is an anti-inflammatory molecule. Again, it's orally available, very stable, and this reduces inflammatory responses like a steroid, but it's not steroidal. So we think it has a number of advantages over steroids, and it can reduce TNF-alpha, reduce the damage which TNF-alpha does, as shown here in some studies we've done in blood. Reduces TNF-alpha, reduces the damage which chronic TNF-alpha reduction does, but it doesn't have the same. It's not going to have the same side effects as steroids, which can't be used over a very long period. We've also done studies with osteoarthritis. Now 2 studies showing that the cucumber extract, standardized to over 1% IDO-BR1, gives a marked improvement in patients with osteoarthritis. It gives a much better response over time than glucosamine and chondroitin, for example, which are often used for trying to control this painful problem. And if you notice from the green bars, there seems to be a steady improvement, which is still improving at the end of this first study, which is 180 days, which does open up the possibility that you actually may have some which is a cure for osteoarthritis here by preventing the damage which is done by cytokines produced chronically, such as TNF-alpha in the joints. And we're working with an American company to develop a standardized cucumber extract, which is called Q-actin. And we've also shown that it works in dogs, for example, as well. So finally, we're primarily interested in active molecules then, but primarily amino-sugars, but not just those, but really we have a niche area in looking at the water-soluble components of plants and microbes. And we're looking to develop a number of products for improving health where these compounds are standardized. The analysis is quite difficult. Many labs, as I said, find working with water-soluble compounds quite difficult. Many of the standards the molecules we've discovered are not available commercially, but we think there's a great opportunity to develop water-soluble extracts which are non-toxic, as standardized herbal products or food supplements which could be used around the world. And many of these could be sourced from Africa and related to the traditional uses of those plants. Thank you very much for your attention. That's the final slide. So I'll stop sharing the.
Thank you very much, Robert. Yeah, very interesting work. I think that, so I'm glad that you accepted the invitation here so that we can more widely talk about this group of compounds. We have some comments or questions. Please feel free to unmute and just ask or make a comment. Stanley.
Hi, David.
Hello.
Yes. Hi. Yes. I think it was a comment. Are these compounds easily isolated, or they actually need advanced equipment for isolation and purification?
In general, the isolation technique for purifying these compounds is using ion exchange chromatography. Because they have a nitrogen, you can separate them away from the ordinary sugars, for example, by attaching the nitrogen to a charge group on an ion exchange resin. That's the best way of purifying them.
Okay, and I think the other one was just a comment in that the ones that you found, that you could give at high doses to cancer cells without having side toxic effects. I thought that was quite interesting because anti-cancer compounds are always associated with toxicities.
Yes. I mean, I think it's a very interesting new approach to finding cancer drugs, but actually it's more difficult because the standard method of finding cancer drugs in the past has been screening compounds on cancer cell lines and seeing which compounds are cytotoxic and selectively more cytotoxic to the cancer cells. But the amino-sugars are not cytotoxic. So in fact, we tried them on a number of cancer cell lines early on and there's no effect. But in vivo, they do have an anti-cancer effect. So many anti-cancer drugs can be missed by doing the wrong assays.
Sure. Yes. So I guess they're acting like immunotherapies, and we are increasingly talking about immunotherapies as new therapies, but what these compounds are doing is potentially that, right?
Yes. So I think they're potentially more interesting than the new immunotherapies which are being developed, which are often antibodies, which are used typically to remove inhibitory signals which stop the T cells, for example, responding to cancer cells. But there are a number of reasons why these feedback mechanisms are there. And there are a lot of side effects caused by the immune therapies because you remove the feedback mechanisms and the T cells can go crazy and start attacking normal cells as well as the cancer cells. So many of these new immune therapies actually cause quite significant side effects by damaging normal tissues. Whereas our molecule is actually activating the T cells, but it's not inhibiting the negative feedbacks, which means that there is still the control mechanism there, so that this should be a safer way of actually activating the immune system than using antibodies, which are completely knocking out probably very useful feedback mechanisms, which are there for a purpose.
Okay. Professor Nyazema, you have a comment? You want to make that? So I think the comment is then around the use of aqueous extracts. Yeah. And Stan, you might be interested to know that we actually, well, I was involved in some work with what you're going to talk about next, compritaceae, but with aqueous extracts. So these compounds would also occur in compritaceae. Yeah, and Dr. Manikandan, I think the question there is, do we have a particular genus where Imino-Sugars occur, or they just occur across? I know you've seen them in pumpkins and in
Yeah, yeah. I mean, then they don't seem to be in every plant you look at. I would say they're in somewhere between 30 and 50% of plants you look at. They are common in particular families, so Leguminosae, and particular genera of Leguminosae seem to produce particularly large amounts, particularly in the seeds, but also sometimes in the bark. And some plants never seem to have them, but you do get some. They do crop up randomly in other families, such as Moraceae. Mulberry seems to be the only species in the Moraceae which contains amino-sugars, for example, but it contains then very high concentrations. So you never know where you're going to find them. It's worth looking at any plant because they could well be there even if they haven't been discovered in that group of plants before.
Okay, thank you very much. I think we'll move on then to the last speaker. So Stan, yeah, you'll tell us the importance of compritaceae, which we see all over Southern Africa and is one of the most beautiful, I think for me anyway, genus of plants, but also very important in traditional medicine in Asia and in Africa. So over to you then.